All right, it's 1 o'clock, so we're going to go ahead and get started with our webinar. Thank you for joining us, and today's webinar is a product of the Sustainable Spotted Wing Drosophila Management Program, supported by the USDA NIFA Specialty Crops Research Initiative. And this webinar will be covering monitoring and sampling tools to improve spotted wing Drosophila management. This webinar will last about an hour, and following that, we will have time for questions. However, you are also welcome to type your questions into the Q&A box at any point during the session, and we'll answer those as they come in. A recording of this webinar will be posted at our project website, swdmanagement.org, by the end of next week. And so if you want to refresh the content or share this information with anyone else, that's where to look. We don't have pesticide credits available for this session, but in some states you can file requests afterwards. And if you would like to do so, go ahead and send me an email for documentation of your attendance. As I mentioned, if you have questions during the webinar, you can type them into the Q&A box and we'll collate and answer those questions. After the webinar, you can send questions to me via email at the email address indicated on the slide. Our presenters today include myself, Hannah Burak. I am currently based at NC State University and will soon be moving to Michigan State University. Phil Fanning, who's based at the University of Maine. Cesar Rodriguez-Sona at Rutgers University. Rufus Isaacs at Michigan State University, and Vaughn Walton at Oregon State University. And these are all members of our Sustainable Spotted Wing Drosophila Management Project team. Today, we will be covering several topics. After I conclude our introductory section, Cesar will take over and highlight adult trapping methods and advancements. Rufus will cover fruit sampling methods. And then Phil and Vaughn will share some real world applications of the knowledge that we've generated with respect to spotted wing Drosophila monitoring and sampling. And finally, I will wrap us up at the end of the hour. As I mentioned, this is a product of our Sustainable Spotted Wing Drosophila project. Our project director is Dr. Ash Sayal, who's based at the University of Georgia. We have collaborators from around the country, including folks based at Washington State University, University of California, Davis, UC Berkeley, USDA ARS, Cornell, Oregon State, Rutgers, University of Maine, MSU, and NC State University. This is a 4-year project with the goals of assembling teams of grower influencers to implement best management practices for spotted wing Drosophila, developing decision aids for farmers to increase profitability, implementing sustainable alternatives to insecticides, reducing the risk of insecticide resistance, and disseminating these recommendations. And the material that we are presenting in today's webinar falls under the development of decision aids, as well as disseminating recommendations that help producers better improve spotted wing Drosophila management. All right, I'm going to turn it over to Cesar, who's going to focus on our first topic, adult trapping methods and advancements. All right, Cesar.
Thank you, Hannah. Hi, everyone. So today I will cover an update on current knowledge on trapping for spotted wing Drosophila and some of the future directions. I want to start with a question: what do we want in a trap? When monitoring for an insect, we want 5 characteristics in a trap. We want a trap to be effective at capturing our target insect. We want the trap to provide us with early detection, a trap that is selective and reduces captures of non-targets, a trap that is easy to use, and also a trap that provides us with some correlation of crop injury. And next. So in an early multi-state study, we compared the efficacy of different lures and baits for capturing spotted wing Drosophila. And this study was conducted in multiple crops across 10 different states and compared the efficacy of 6 different treatments, as you can see here. This included apple cider vinegar, which was the bait that we originally used when spotted wing Drosophila first invaded the US, yeast and sugar, a fermenting bait plus apple cider vinegar, a Drosky drink, and 2 treatments with a synthetic lure, one with and one without apple cider vinegar. Next. So here is the composition of these baits. This work has been published, and you can find this information in our publication. So I'm not going to go into too much detail on what it was in each of them, but you can see that a lot of all of them contain fermenting products. Next. So when tested for efficacy, we found that the fermenting bait and the trestle lure over the apple cider vinegar capture the highest number of flies across all crops. Next. When we looked at the sensitivity of these traps, these baits and lures, we found that all of the new baits and the new lures capture flies 1 to 2 weeks earlier than apple cider vinegar. However, the attractants that we tested were most effective at capturing flies were also the least selective. The fermenting bait and the trestle lure over the apple cider vinegar, as you can see, capture more non-targets than the other attractants that we tested. Since that work was completed, several baits such as Suzuki trap and Drosa lure, and also lures such as the trestle lure and the century lure have become commercially available for monitoring spotted wing Drosophila. Both the trestle lure and the century lure are based on volatiles emitted from fermentation products. Next. So since the current lures are based on fermentation volatiles, and spotted wing Drosophila is known to utilize various odors, including odors from healthy fruit, yeast, and leaves to locate mates and food and oviposition sites, we tested whether combination of these odors would increase spotted wing Drosophila captures. For this, we combine fermentation odors with odors from yeast, with odors from leaf, and also with odors from both yeast and leaves. We found, however, that adding odors from yeast and leaf to the fermentation odors reduces spotted wing Drosophila attraction to the fermentation odors. And this happened both during the season as well as during the off season. So these studies also show that the flies during the growing season are highly attracted to fermentation odors, but not to yeast and leaf odors. However, after the season, when the number of fruit available on the plants was low and the leaves were senescing, the attraction to yeast and leaf odors increased, indicating that the response of these odors is affected by the odors in the background. So far, lures based on odors from fermentation products are the most effective at capturing spotted wing Drosophila. However, as I mentioned, these lures are not very selective, which makes processing of these traps very difficult and time-consuming. Thus, we have been asking whether we can make these traps more user-friendly. In a recent study by Kirkpatrick and colleagues, tested different trap designs. They tested a combination panel that consisted of a red sphere in a yellow background, a red sticky panel, our standard liquid trap, and a white, yellow, and green sticky panels. What they found was that the red panel and the combination panel captured most flies. And this agrees with other studies that find that the spotted wing Drosophila is attracted to red. It also shows that we can place the liquid we can replace the liquid trap with a red sticky panel to monitor spotted wing Drosophila. So the use of these red sticky panels makes it easier to process the traps, since traps can be checked directly on farms. In fact, male spotted wing Drosophila that have the spots on the wings can be easily identified on these traps, and that can provide a quick assessment of their presence in the fields. So the study I show you was done in one state, in Michigan, and in few crops. Thus, we wanted to test if these traps, these red panel sticky traps, can be used in multiple crops and multiple states. For this, we conducted a multi-state study in 2018 to compare the liquid standard traps baited with commercial Sentry lures with a red sticky panel also baited with a Sentry lure. So this study was conducted across 5 states and was conducted in high bush and low bush blueberries, raspberries, and blackberries. So in all these graphs, I'm showing shows the abundance of spotted wing Drosophila across the season. And in blue, I'm showing you the spotted wing Drosophila counts in the liquid traps, while in red lines is the captures of spotted wing Drosophila in the red sticky cards. In general, what we find, for example, in Oregon, is that the liquid traps in blue capture more spotted wing Drosophila than the red traps. So you can see that these are the results from Oregon. Next, these are the results from New York. Next, and New Jersey. And next, Maine, low bush blueberries, and North Carolina blackberries. So you can see that in general, we find that the liquid traps capture more spotted wing Drosophila than the red panels. However, their efficacy seems to vary across crops and states. In general, the red sticky traps work better in high bush blueberries than in raspberries and blackberries. So this year, a similar study was conducted, but this time we included more states. We included at least 10 states, and also was done across different crops, blueberries, blackberries, and hazelnut. And we compared the efficacy, sensitivity, and selectivity of the liquid traps versus the panel, the red sticky panel traps baited with Sentry lures and also Trece lures. So on the left of these graphs, we show the results from the liquid traps, and on the right, we show the results from the red panel traps. In red are the data from the Sentry lures, and in green and purple are the results from the 2 formulations of the Trece lure. So in Florida blueberries, the panel traps capture similar numbers of spotted wing Drosophila as the liquid traps, as you can see. This was the same in Georgia blueberries, but interestingly, in Maryland blackberries, the red panels outperform the liquid traps. So they capture more spotted wing Drosophila. Next. In New Jersey blueberries, the red panels also outperform the liquid traps as far as the spotted wing Drosophila captures. And similar results were found in Michigan in blueberries. The spotted wing Drosophila captures were lower in Virginia blueberries in the red panels compared to the liquid panels, and the liquid traps outperform the red panels in low bush blueberries in Maine. So when we looked at the selectivity of these traps, these are the captures of males in the liquid traps during the first half of the study, the first 5 weeks, and also on the second half of the study from weeks 6 to 10. And you can see that males were captured on liquid traps on the first week of sampling. So when compared to the liquid trap, the red panel traps capture the first spotted wing Drosophila males at the same time. This is important since, as I mentioned earlier, we're interested mainly at the possibility of monitoring males on the traps, since they are easier to identify than the females because of their spots on the wings. So this is similar data, but this time for the females. And females were also captured on the liquid traps on the first week of sampling. And these data were similar for the red panel traps. Spotted wing Drosophila females were also captured on the red panels on the first week of sampling. So when we looked at selectivity, these are the proportion of spotted wing Drosophila females in blue, males in red, and non-spotted wing Drosophila flies in green across the different lures in the liquid traps. As you can see, selectivity was less than 50% for all the treatments. And these results were similar for the red sticky traps. Although one of the Trece lures in both cases was more selective than the other lures. This lure, the Trece HS, captured more spotted wing Drosophila, especially it captured more females, as you can see in blue. So another important aspect of a good trap is whether it can provide information on crop injury. Thus, we conducted a study this year to determine whether spotted wing Drosophila captures on red sticky panels correlate with fruit infestation. This study was conducted in 3 different states, in New York, Georgia, and North Carolina, and in 2 different crops, in blueberries and blackberries. It was conducted at each site. They placed 10 red sticky panels baited with Trece lures, and fruit infestation was monitored 1 meter from each of the traps. So these are the data from blueberries in New York. The graph shows the cumulative male trap captures during the season in black and the fruit infestation in red. As you can see, as the trap captures increase, the level of fruit infestation also increased. The graph on the right shows the overall correlation between trap captures and fruit infestation. In general, trap captures positively correlated with fruit infestation. And from this data, it was found that each spotted wing Drosophila male capture on the trap translate to 1 egg per 120 berries nearby. So these are the same data for blueberries in Georgia. Here, the pattern was not as clear since the level of infestation, as you can see across most sites, was fairly low. And for this reason, the correlation was also not as clear. And these are the data for North Carolina blackberries, and similar to the data from Georgia, the correlation was between male catches and fruit infestation was not as clear. And also, as you can see, the correlation was weak. So in conclusion, what we have learned so far is that the fermentation lure remains the most attractive lure for capturing the spotted wing Drosophila. And adding other odors lowers its efficacy. We're also showing that red panel traps can be used instead of liquid traps for monitoring spotted wing Drosophila. However, their efficacy varied by crop and lure type. It seems to work better in high bush blueberries. We also found that the red panel traps provided similar early detection as the liquid traps. However, selectivity of the traps still remains low. We also found that spotted wing Drosophila captures can be correlated with fruit infestation, at least in one site in New York. So next. So for future directions, we have demonstrated so far that the red panel traps can be used for monitoring spotted wing Drosophila, at least in some crops and regions. We still need to improve the selectivity of the lures to reduce captures of non-targets. And research needs to be done on the development of action thresholds. Our team also plans to conduct an economic analysis on the benefits of using the red sticky panels. And finally, there is also the possibility of using automated devices for monitoring spotted wing Drosophila. In fact, some of these devices are being developed and are currently being tested.
Thank you, Cesar. Now we're going to move on to discuss fruit sampling methods and how that information can be used to improve management with Rufus.
Okay, thank you very much, Hannah. Hello, everybody. Thanks for joining us today on this webinar. So I just wanted to give a little background about the value of sampling in different ways for making decisions for spotted wing Drosophila management. We've just heard all about traps, so I don't need to spend too much time on this, but they do have some issues with not being selective. Other flies are caught. And although we're trying to find ways to improve correlation to the risk of infestation, at the moment we see, as we just saw in Georgia and North Carolina, not always really strong correlation to infestation risk. Egg monitoring, we'll hear a little bit more about later, but this is another way that we could look at this. And of course, the eggs are laid before the larvae, so it might be useful to know about egg laying, but that is time consuming, requires a microscope, they're very small, etc. So I'm going to spend some time today talking about larval sampling and the methods that have been developed through this project to be able to sample marketable fruit, provide information on the need to spray, and hopefully make good decisions about spotted wing Drosophila management in farms. Next slide, please. This approach was developed with a collaboration with North Carolina State University and with my lab, and a lot of the credit goes to Steve Van Timmeren, who was the lead author on this. The basic approach is collecting berries from fields. We put those in a plastic container. You can do a squeeze with your hand, or in this case, you see them doing individual berries. Put that in salt water, and then this filter with some very coarse mesh sorts out the berries, and then the liquid flows through a coffee filter that's at the bottom and goes into the sink. So that leaves you with, if you can go to the next one, please, Hannah. That leaves you with a count of spotted wing Drosophila larvae per sample. And so these numbers are rather high because they're from fruit that we deliberately infested to be able to test these different methods. In the graph at the top, we've been trying to improve this method and just see whether it can be done more quickly, more efficiently. So one thing we looked at was fruit squeezing, which is the black bars. The legend got cut off, so my apologies for that, but the gray bars in that top one are where there's no fruit squeezing. So we're comparing with and without fruit squeezing. And that didn't make a difference for medium larvae or large larvae. There was a little bit better ability to find the really small ones, I think because the squeezing helps them get out of the berries. In the bottom graph, we see that salt water is in black, brown sugar water is in the gray, and then the lighter one that was also cut off is just clean water without any sugar or salt in there. And interestingly, those gave very similar results. I think because usually salt water or sugar are used to make the larvae float by making a higher density solution. And when you're sorting through a filter, you don't need that. So I think we could dispense with salt water and brown sugar, or brown sugar, if we're doing this through a coffee filter. Next slide. So this is what you end up with looking into the coffee filter. You can see the larvae there in the bottom of the container. It could be with a hand lens that you look at those and observe and count whether you have larvae there. We often use a microscope. You can go again, Hannah. And we've counted, Steve's counted the amount of time it takes to run these samples. So this is the putting the fruit into the liquid and then running it through the sifter and then the counting. So on average, it's 2 to 4 minutes per sample. You, of course, also have to build in the 30 minutes in the liquid if you do that, and the time sampling. Practically, a lot of people that are implementing this are taking those samples in the field, putting them in the liquid into containers in the back of the truck. By the time you get back to the office to sort through them, that 30 minutes may already be done. Next slide. Another piece we've looked at here is the likelihood of getting a spotted-winged Drosophila out of some different methods, again, to compare the filter method that we've modified with the adaptations I just described compared to the standard one. So those are the two greens. And in here, we see very similar levels of spotted-winged Drosophila larvae per gram of fruit. You'll notice that we're only picking up about probably 30 to 40% of the larvae that were in there with this method. So that's something to be aware of, that you may not get them all, because when we hold the fruit and the flies emerge, you see much bigger numbers. And then you also see the brown sugar flotation on the right there that just indicates that that's not as effective. Okay, next one. So this summer we've been doing some comparisons of spotted-winged Drosophila per gram of fruit and also the detection likelihood. So that's really important. It's not just the number of flies or larvae that you find, but are you just as likely to be able to detect that it's present or absent? And so the table on the left shows that we have very similar numbers through the season from, in this case, this is at a research station, so it's not a managed field, not a commercially managed field. But there at the beginning of July, we're seeing as the fruits start to ripen, 5% of the samples have some level of larval infestation, and then that climbs through the season, and it's very similar numbers on the right with the modified test. So that's encouraging that it can do a similar job. But when you do look at the number of larvae, it's also fairly similar through the season. Okay, next one, Hannah. Thank you. And so this has been implemented in some commercial farms where we are trying to bring fruit sampling into the decision-making at commercial farms. These yellow dots show where we might sample around a farm, especially up against wooded areas in our region. That tends to be a source of SWD. And then we also want to know about the middle and the other edges of the fields. So I'm going to run through a series of slides here where you can see this example farm where we had early, middle, and late harvested fields. And then we're starting in the middle, at the beginning of July. July 5th was this sample. And you can see that at the beginning, these early harvested varieties, for our region, early harvested blueberries typically miss a lot of the SWD pressure. So we've got 0 larvae per sample of 3 ounces of berries. And then, Hannah, as you progress through these slides, you'll see those numbers change. So we go to July 12th, a week later. We did pick up our first fly at this farm, happened to be in the middle of the field, or the farm, but still 0 larvae in the early harvested berries that are just starting to get ripe. And the middle harvested ones were still green, so those didn't yet need protection. You go ahead a week, and you start to see the counts coming up in the early harvested variety here and some detection at the borders. So this can help a grower think about where protection is needed and which fields need to be sprayed. And you can go ahead, Hannah, through the sequence there, and you see the same kind of thing progress with through August with more pressure at the edge and less on the inside. So now we're into the later part of August, and that early harvested block is already done, so we're not sampling there anymore. And now we get the really higher pressure that shows up as we get into the late part of the season, and these fields stop being harvested. They go post-harvest, And especially when the growers stop the sprays, you start to see these much higher numbers. So the last slide on this is just to reiterate that I think these harvest decisions can be guided by this kind of sampling. We can use the traps to tell us when spotted-wing Drosophila gets active, but it doesn't tell us what's going on in the fruit. And so this can guide when to harvest, how many times you're going to pick a field. It can guide insecticide treatment decisions, so the ones that I show there. And then I can't see the last one because all the question boxes are up, but whatever it says at the bottom about spotted-wing Drosophila control. Let's see what my slide says. Oh yes, confirm effective spotted-wing Drosophila control. So I focused on the detection of larvae, but I think it's also really important for growers to know that there aren't larvae there and know that those fields can be harvested and marketed. So that's the end of my section, and I'll turn it over to Phil and then Vaughan. Thanks.
All right, so next we'll be moving in to talk about some on-farm examples of utilizing these different monitoring and sampling tactics. First, we'll discuss work that's been done in Maine, and Phil Fanning is going to lead that discussion.
Hey, thanks, Hannah. So if you just progress to the next slide there. So here in Maine, we monitor with a yeast sugar solution, and we use those red solo cups that you can see in the picture on the left-hand side. We recommend the growers put out 3 red solo cups per site, or per area of their field that they're monitoring in, and they get checked weekly. And through time, we have also been conducting that monitoring and also looking at fruit infestation adjacent to those traps. Based on 5 years of data, we developed action thresholds, and I'm saying we, but it was my predecessor, Frank Drummond. So we were able to look at our trap captures and correlate that to our fruit infestation. So our trap captures are based on males, mostly because males are easy to spot with those spots in the wings. And how it works is you get a cumulative average for the number of males per those 3 traps, and then that would correlate to the possibility of not having or having infestation in your fruit the following week. So if you look at this table on the right-hand side, you can see if you had an average cumulative male trap capture of 3.5 males, that would mean you have a 90% chance of not having infestation, but a 10% chance of having infestation that following week. So that's a threshold that's commonly used here is 3.5 cumulative average males. On the next slide, we see basically how we can use that information and how we can adapt it. So as I mentioned, this action threshold was developed by my predecessor, Frank Drummond, here in Maine. And then in collaboration with Miguel Gomez, who's also a PI on this grant, they were able to develop a decision aid tool that is easily accessible to our growers here in low bush blueberries that's based on Excel. In that, they can use those cumulative male trap captures. They can input them along with some other characteristics of their crop, how much they anticipate, the control costs would be if they were to spray, the number of acres, the price of their farm, and they can use that then to make decisions on whether it is economical to make an application for spotted-wing Drosophila, or if maybe harvesting earlier to avoid making that application makes more sense economically. So it's a really good tool, and it's one that's been very accessible to growers here. On the next slide, I'm going to talk a little bit more about our work using the filter method that Rufus just introduced you to. So we know that basically spotted-wing Drosophila can be pretty picky about where it lays its eggs. You can see here on the left-hand side a really nice graphic by Marco talking about how spotted-wing Drosophila uses the canopy of high bush blueberries, and we've noticed through time, and Lauren Dittenbrock did some of the earlier work on this, showing that in that interior of the canopy where the conditions are favorable, that's where we get the most egg laying associated with spotted-wing Drosophila. In the eastern United States, traditionally we would have dealt with blueberry maggot fly. And blueberry maggot fly is active, and the infestation associated with that pest has always been around the perimeter of the field, especially here in Maine and low bush blueberry. And our early work in recognizing that has, drove us to doing management for that pest targeted around the border or the perimeter of the sprays. And that significantly reduced the inputs and the costs associated with managing that pest. So in recent years, we have been tracking the infestation associated with spotted wing Drosophila. So on the next slide, you'll see basically how we do this. We go out into our fields, we measure in transects from the edge of the field into the interior of the field. We do multiple transects per site, and we monitor at fixed locations all the way up to 160 feet into the field. At each location, we collect 3 6-ounce samples, and we do the salt flotation method to look at whether we have infestation within those samples. So on the next slide, you'll see basically some of the results of these. So this is our first site. This data is from last year. When we get all of that infestation data, we can produce these heat maps to indicate where the distribution of infestation is. So on this map here, you'll see basically, the distance between our transects, the distance into the field, and you'll also notice these different colors down on the right-hand side. So blue would indicate that you have low levels of infestation, moving up to red where you have high levels of infestation. So this is our first sample at this site. You can see that for the most part, it's mostly blue. There was really low infestation. There might have been 1 larvae in 1 sample. If you move on to the next sampling date on the next slide, you will be able to see that here we started to see more infestation. I should mention this is a completely untreated site. So this site was not treated by insecticides. You can see that red pattern coming up at the edge of the field within the first 40 feet. When you move on to the next slide, which is the following week, this was the week where we finished the trial. You can see basically that most of that infestation is centered around the border of that field, and again within the first 40 feet. I put a little circle around the figure there on the right-hand side, the 25. So that's the maximum number of infestation we had associated with a sampling location. In the next slide, in the next field, we're going to look at basically a different site. So this was similarly, it was an untreated site. Oh, there you go. This is just one snapshot. So this is another site. You can see here, our top here was 14. So we had different pressure at this site. It was less than the previous site. But again, we can notice that all that red pattern is associated with the edge of the field. Then the next slide is our third site that we're highlighting here. So this was a treated site, and this was basically the last sampling at that site. The grower had already stopped spraying associated with spotted wing Drosophila because of the pre-harvest intervals in those sprays, and some infestation had snuck in. But for the most part, again, it's really low levels associated with those treatments, but it's centered around that border area of the field. So when we visualize that, completely, we can see here in the graph on the right-hand side, this is basically the spatial distribution of infestation from spotted wing Drosophila moving from the edge of the field into the interior of the field, or into 160 feet into the field. This graph shows the mean proportion of SWE infestation. And what's pretty clear, I think, from those heat maps in this graph is that there is a clear perimeter-driven behavior associated with spotted wing Drosophila, at least here in low bush blueberry and at least early in the season. What all of those samples came out to across 2 years, and we have a third year to add, is about 60 to 70% of our infestation occurs within 40 feet of the field edge. I think it's 90% occur within the first 100 feet. So that's a real potential to overlap some of our management for blueberry maggot fly at the perimeter of the field and just focus on that at least earlier in the year. Moving forward, we're thinking of an approach where we will monitor infestation at 10 feet and at 120 feet into the interior of the field. So we'll have our traps to initiate when we think we might be getting infestation. Then we can monitor infestation to make sure our perimeter sprays are working. And if we potentially notice that we have some infestation on the interior of the field, that's when we might focus our management across a wider area of the field. And this could be a really good way of reducing our inputs associated with controlling spotted wing Drosophila. I think that's my last slide, so I'm going to pass you to Vaughn now.
All right, thank you, Phil. Vaughn is going to take us out onto the West Coast and share what they've been implementing with growers in Oregon. Take it away, Vaughn.
Thanks, Hannah. This is work also in collaboration with Dr. Jana Lee, who is providing some additional funding through an area-wide grant. So I'm going to be presenting data over 2 seasons. If we can have a look at that next slide there, just to kind of give us an idea of the life cycle. This is just a little bit of education, which most people don't need at this stage. But when we're looking at Drosophila Suzuki life cycle, really important to note that the period between egg laying and then larval development depends on temperature. So if we go to that animation in this slide, I think there's a time period in there, anything between 6.8 and 14.7 days. Now, as Rufus very clearly stated earlier on, really difficult to see eggs. However, eggs are laid a while before we can start seeing those larvae. And in many of the regions here, if we have that little bit of extra time, that can help us decide as to when we put some management strategies out there. So if we look at the program that we used over the past 2 years in the next slide, this is a reasonably big area. This is an example of one of those fields. You can, when you look at these rows here, the right-hand side of the row is the east side, left-hand side is the west side. I took a photo of some berries there. You can see shading. This, the collection of the berries were done on the east as opposed to the west side of the canopy, because we believe that it's slightly cooler there and that the berries are more likely to be infected. In the center of the canopy, on the east side of that row, we looked at 4 locations overall during the 2 years, a total of about 850 acres. We did similar sampling as to what Rufus and Phil did in their experiments, 16 to 36 samples, 10 to 20 berries per sample. And then we went every second day, a total of 150 dates, about 37 some dates per location. Quite a few samples, about 4,600 over those two years, about 57,000 berries that were looked at under the microscope. Quite a lot of berries to look at. If we look at the next slide here, just to kind of give you an idea of what the sampling methodology looked like. So we did our 12 sampling sites. 4 of those, 4 or 3 of those, depending on how you look, were from the perimeter of the field. And so we were separating those and were able to look as to where that infestation happened. If we look at the next slide, kind of giving you an idea of how we process those samples. We would then bring those samples back into the lab, look at them, individual berries under the microscopes, both for eggs and larvae, and we would note those. After that, we would take those berries, we would constitute a single bag and then do a modified fruit dunk method, kind of similar as to what Rufus explained, slight modifications to those because these are in commercial settings. Also, in addition to the fruit dunk method, all of the data, whether it's egg data or larval data, were then sent to the grower within a 24-hour period, allowing them to make some kind of a decision as to whether they should manage or not. So if we look at the next slide, just kind of as an example here, you can see here, these are all our sampling dates in the graph on the left there, and you can see these little bubbles. This is a late variety, or a variety that ripens relatively late, up to the latter portion of August. This is legacy. It ripens also over a lengthy period of time. And so when you look at those bubbles, you can see 3 little bubbles there. Every time that number was brought back down by a management action by the grower. What we learned from these samples also is that very late in the season, it's virtually impossible to control those spotted wing Drosophila numbers in the fruit. And you can see how the numbers blow up really late in the season. This is a phenomenon that's been seen all over the world, many locations. You can go from very low infestation, 0 to about 80% infestation within a 1 to 2 week period. So really important, especially late, as was indicated by both Rufus and Phil. That's the period that we need to really look into. When we look at the distribution of those egg laying, if we look at that animation in this slide, we found similar to Phil, sorry, one more, similar to Phil, that about 90% of all positive samples were always found on the edge. Now, there is some really good work done on fruit firmness, initially started by Dr. Burak. Our data showed that there is a real clear correlation also in terms of ripening when we're looking at that very, very late period of the season. But key here is to see that about 90% always are found in that outer 40 feet within the field itself. If we look at the samples themselves on this next slide, looking at the sampling dates, about 150, as we said earlier on, and this is where the differences start showing up. When we look at that 150 samples, about a third of those sampling dates, we found eggs. We found in about a tenth of the samples larvae. So 17 dates we found larvae, about 48 dates we found eggs. Pure total number of egg counts versus larval counts, about 4 times more eggs than larvae, about 4 times higher mean egg counts than larvae, more or less the same as the line just above that. So next animation in this slide here is the average days of larval count after egg count. And if you take all that data, all of those data points together, we're finding that by doing egg counts, you're able to find those first counts about 10 days before you start finding larval counts. And this is, I think, of real value to our growers here, because you can find those eggs first, you find the larvae much later. Now, the next question to ask here is, who's going to be counting eggs? Many of us think that it can take a lot of time. Are the advantages more than the disadvantages or not? So this is why I have that next slide up here, which kind of gives us an idea of the comparison of those methods. 3 methodologies. We have our modified fruit dunk at the top there, and then the corresponding numbers directly next to that. We had the separate bag microscope methodology that I explained a little bit earlier, just below that. And then when you're taking a composite bag and you're looking at a composite bag, you're taking all 12 of those bags. You're only selecting a sub count of those berries from that bag right at the bottom there. And so generally speaking, when we're comparing those 3 methods with each other, the disadvantage of the modified fruit dunk is that we're not seeing eggs, as you can see in the middle here. The advantage here is that it's really quick. It's only taking about 6 minutes to take those samples. If we compare that with our separate bag microscope, it's about a quarter of the quantity of time. And so many of us will say, well, you know, the advantages of doing the modified fruit dunk outweighs the separate micro bag methodology. Our growers worked with us and came up with this composite bag micro methodology, where you're only selecting a certain number of berries out of that bag. We can drop that time used from 20 to 10 minutes. But the advantage that we get for the sample analysis, but the advantage we get out of that is that we can see both larvae and eggs here. And so, you know, the advantage of being able to count the eggs is it gives you a few days extra that helps you time your sprays a little bit better. So just quickly looking at the advantages and disadvantages, and I know I have about a minute left here. Modified fruit dunk, really quick. Disadvantages here, no eggs or location of where those eggs are. But what we're seeing now from all our data and all these regions are that, you know, we're finding the majority of the eggs on the edges of the field, and we can assume that. When we're looking at the separate bag micro, not everyone's going to be able to do that method, but we can find eggs, which buys us a little bit of extra time. When we do a composite bag micro method, we can reduce that time, still be able to count our eggs, which buys us a little bit of time. So this is a method that our growers seem to think that provides a lot of value. The big thing is that it's time consuming, but it's really useful in terms of making sure that we have a good quality crop. And I think that's about it that I have at the moment. Thank you so much.
All right, thank you, Vaughn. It's really useful to see how this information that we've generated in field-based research experiments is able to be translated to actual applications with our grower cooperators. And it's really nice to wrap up our set of slides with that topic. I wanted to provide you guys with a few take-home messages as we conclude the research component of today's webinar. First, to go back to Cesar's points about adult trapping methods and advancements. Those adult trap captures may be useful in some cropping systems to time start of treatments, and the lures and trapping systems perform similarly with respect to trap timing, which means that our red panel traps, which are easier and faster to service, may provide a equally useful timing assessment. As Rufus indicated when he discussed fruit sampling methods, fruit sampling is an important part of spotted wing Drosophila monitoring and provides a valuable additional piece of information to adult trap captures and should be part of growers' practices. And then as Phil and Vaughn indicated, in blueberries, infestation appears to be higher on field edges, and these fruit sample programs are able to improve management outcomes for stakeholders. However, we have done most of this work in blueberries thus far, and so we need to expand these applications on farm to crops beyond those systems and throughout our national program. I want to turn it over to Cesar for just a minute because we would be remiss if we didn't acknowledge a colleague who contributed greatly to the work that we presented today. Cesar?
Thank you, Hannah. We wish to dedicate this seminar to the memory of a great colleague and friend, Dr. Larry Good from Michigan State University, who passed away earlier this year. Larry made extensive contributions in the field of integrated pest management in fruit crops, and in particular for his work in the development of better monitoring techniques for spotted wing Drosophila.
And I just wanted to follow that statement by Cesar by just letting everybody know who's on the webinar today, in case you hadn't seen this, that our department, where Larry was the tree fruit entomologist for more than 20 years, in his memory, we've established an endowed scholarship. So there is a fund to support students. He was a great supporter of students. And so that QR code there takes you to the particular page if you're interested in contributing to this. But the goal here is to support future students in our department that are trying to get to conferences or establish collaborations with other researchers, because that's something that Larry was definitely a strong proponent of. So this is to honor his memory and also continue the work that he started here. Thanks, Hannah.
Great. Thank you, Cesar and Rufus. And as indicated, Larry is truly missed both in the spotted wing Drosophila world and more largely in the entomological community. Before we conclude today, I want to remind you of our spotted wing Drosophila resources. We have a number of regional websites where information on spotted wing Drosophila management is available. Some of the personnel who are associated with these regional resources are on our webinar, so it's nice to see your names popping up. Our project website is swdmanagement.org. The webinar recording from today's session will be posted next week at the website if you want to go back and catch additional information or share this with others. And we have some social media platforms as well as blog sites with timely updates on spotted wing Drosophila management. Finally, we do have about 5 minutes here before the end of the hour. So I want to give folks a chance to ask additional questions. You can put those in the chat box or the Q&A, and we'll address those for the next 5 minutes over the audio. Although we did have one topic that came up a couple different times. So I want to kick us off on that topic, which is ground floor management. Vaughn, maybe you can touch a little bit on the impact that what's happening under the canopy can have on spotted wing Drosophila infestation. This came up in a few comments in the chat.
Yeah, so when you look at overwintering, and this is kind of related to weeds and so on as well, but when we're looking during the overwintering period, we had one study a few years back where we were trying to figure out the overwintering of the flies a little bit better. And what we found out from that is that these bugs cannot survive for more than a 1 week period if they don't get sugar. And so where do they get that sugar during the winter period? They survive as adults during the winter period, and they need that sugar for sustenance. They could get it within the blueberry field itself as well, but those sugars during the winter period is much less available. When you look at the leaves and needles dropping onto the ground, during the winter period, you have a nice thick layer there with microorganisms proliferating on those. And what you get from those microorganisms is an adequate quantity of sugar, which then we believe, in addition to many other sources, also help support those individuals during the winter period. We don't really see that in hazelnuts. There was a question about hazelnut. Hazelnut floor management is really important. We don't really see that in blueberry or anything like that. We think that's why it's so important for these bugs for their survival during the winter period. And I know there's a question in the Q&A, why are they not in the field? Why are they in surrounding vegetation? Very often, my best understanding from that is that the surrounding vegetation is a much better microclimate than in the field itself. You have much more sun in the canopies of small fruits, at least. I don't know about cherries, but it's a less suitable environment that we're producing. Some of it might be because of pesticides that are being sprayed, but microclimate also play a really important role.
And just to add to the response to the question in the Q&A about why spotted wing Drosophila are higher on field edges, we've consistently seen that with adults for years. Our trap captures for adults are always higher outside of the fields than they are inside the fields. That has something to do with the fact that we are actively managing them in the field as opposed to not actively managing them in the non-crop areas in most cases. It also, as Vaughn indicated, has to do with the environment and microclimate. I do want to share, however, some observations that we had in blackberries. Different system, different field scale, different landscape architecture around the fields. And in those cases, while we do see infestation pop up on the edge of the field first, we often see infestation rapidly spread to the center of the field within a week or two of detection. And so we don't quite see the same density effect in blackberries as we do in blueberries in other states. And so it's also important to consider what crop you're working in, the time of the year that it's being harvested, and the overall attractiveness as compared to what else is out there in the environment. All right, so I see a question in the chat box. Has there been any research on controlling SWD in small blueberry patches? For example, repellents or other tactics? And I'll say one thing about small scale plantings, and that is that netting can be highly effective, particularly for blueberries. It is a lot of work, but for small scale plantings where you can cover the plants with fine mesh netting, that has been demonstrated to be very effective at preventing infestation from developing. So that is one tactic to consider for small scale home or non-commercial plantings. Do folks have other comments that they'd like to share about strategies that could be used by non-commercial growers? And Cesar and Vaughn just indicated in the chat box. Cesar, if you want to echo that.
Yeah, I was just mentioning that we are working on repellents and deterrents, but that research is still at early stages. So we are still working on the efficacy, especially at field sites, you know, in natural settings. They tend to work in the lab, but not. We need more data in the field.
Wonderful. All right, well, it is 2 p.m. So I'm going to go ahead and conclude the webinar. Please do reach out to the email address indicated on the screen if you have additional questions. We will follow up with those via email, also on our project webpage. So thank you again for attending, and we will get this up and shared shortly.